Good morning or good afternoon, maybe good evening, maybe good early in the morning to some of you because you all have very different schedules. I think I finally got Screencast-O-Matic to work again, and so I'm going to record a lecture on the brain. I've broken up this lecture into at least two pieces. We'll see if it might be three pieces based on how long it takes me to get to some goal points. But I'm going to talk about the brain. This lecture definitely builds on the readings that I hope you've done today. And speaking of that, I peeked at the readings myself today and noticed a pretty big mistake. I think it was in table 6.1 or figure 6.1 when I referred to an image of a sheep's brain that my editors apparently changed. So I don't think I made that mistake myself, but I'm ultimately responsible for it. So I hope that wasn't too confusing, and I hope you still understand that the human brain is lateralized into two symmetrical halves. All right, so let's jump into this lecture, but well, actually, before I do, let me tell you that I've done something that I hope you'll find helpful. At the top screen, I think of every page in this lecture and the next, I put down those 6 key words that I think you saw in the first module on the brain. Brains are conservative, brains are compartmentalized, etc. And after I make a point on each page, I'll make a reference back up to that little header at the top to indicate the gist of the point I just made. And sometimes it makes a couple of points or 3 points, but I think that's a good way for us to think about those 6 ways of thinking about the nature of the brain as we work our way through this lecture, which does overlap, I would say, at least 50% or so with what's in your readings. And I hope for you that's good news. Okay, so we're going to talk about the brain and the nervous system. And the first thing I want to do with you, if I can, there we go, is to share with you an image of a human brain. That's Homo sapiens over on the right, and that's a chimp brain over on the left. Now, a typical adult male chimp weighs about 90 or 100 pounds. A female chimp's maybe 75, 80 pounds. It varies, of course, a bit, but chimps are quite a bit smaller than people. Of course, people in countries that don't eat a lot of food are often 130 or 40 pounds as an adult male, not like the 200-pound American men because we eat so much. But at any rate, we are something like 50% bigger than chimpanzees. And if we were just like them in all ways, except bigger, our brains would be about 50% bigger than their brains. But our brains are much bigger than their brains. Okay, so our brains are at least 3 times the size of their brains. Our brains weigh about 3 pounds. Their brains weigh less than a pound. And so our brains are very different than their brains. On the other hand, if you look carefully here, I think you can see that our brains are also quite similar to their brains in overall structure. So, for example, both of the brains that you see here have something you read about today, I hope, called a hindbrain. That's the very low part of your brain. You could think of it as part of the spinal cord if you wanted to. It regulates really basic things like sleep and breathing. Both brains have that structure. It's very, very similar in our brain and in a chimp's brain. Okay, so I circled it for you there in both brains. Both brains also have a frontal lobe. It's the front quarter, or more like 30 or 35% of the brain, that plays a major role in personality. I didn't mention that in the book, but it's true. It also, of course, plays a major role in things like emotional regulation and risk taking. So both brains have a frontal lobe. There it is, color coded in green for you. Both brains, of course, contain very highly specialized Purkinje cells. That's a certain kind of neuron that are in the cerebellum. They have hundreds or thousands of dendrites that feed into the cell body that allow your cerebellum to help you make some very sophisticated movements. And as you may recall from the reading, it also helps you do a lot of other things that we've only discovered in the past decade or two. So the cerebellum is almost like a little backup brain. It does a lot of complicated things, but people associate it most with helping you throw a spear or do gymnastics or do a complicated uh, procedure when you're jumping rope or something. It involves things like holistic, rapid movements. But both chimps and people have a cerebellum. In fact, all mammals have a cerebellum. And so many brain structures are common not just to people and other primates, but to people and other mammals, and some things, as you know, to people and reptiles or people and insects. Okay, so that's the cerebellum. You can see it there. And finally, both brains use dopamine, both brains use acetylcholine. Our brain uses 68 additional neurotransmitters. I think chimps use almost that many. I haven't read up on that in a while. I'd be surprised if they don't use at least 30 or 40. If you make me guess, I'd say probably 60. There might be one or two that we use that they don't, but our brains are chemically a lot like their brains. There's no doubt about that. Okay, so if you think that I know the chemical composition of dopamine or acetylcholine, no, I haven't had a chemistry course in decades, let's just say. But if you're a chemistry major, you can look at the chemical composition, the formula, the chemical nature of dopamine, and you can look at acetylcholine and know that they're pretty complicated chemicals that do their jobs. Okay, so that's point number one. I hope if you pause to think about that a little bit, you'll see that I just said something about the way our brains at the chemical level, at the structural level, at the functional level are not that different from chimpanzee brains. That makes the point that the human brain and the chimp brain are similar. We share a common ancestor. We didn't evolve from them, but we share a common ancestor. And that makes the point that brains are conservative. And I forget if I didn't circle any, I went a little too quickly there. That makes the point. Now, you could also make the point that brains are complicated, ours more than theirs, brains are compartmentalized, but I mainly make the point there that brains are conservative, okay? When something works, evolution rarely gets rid of it and starts all over again. Okay, let's talk about the brain and the nervous system. And I'll tell you here that as late as the 1700s, so not that long ago, in the grand scheme of human history, nobody really knew how brains work. People thought that nerves were hollow tubes where that air moved through. Maybe water moves through them. They looked around their worlds and looked at this cool thing called plumbing and said, well, when we've seen some brains chopped up, it seems like there are, tubes in there. So maybe it's like a system where things are going through the tube. So really, nobody had much of an idea at all. There were all kinds of guesses. It wasn't until the late 1700s and the early 1800s, way before psychology became a field, that a couple of guys in Italy, Galvani and Volta, and that's the Volta that the Volt, not the Chevy Volt, but voltage is named after. These guys did a lot of experiments with electricity, and they discovered independently, and then they debated it a lot, that nerve conduction is bioelectrical. It sort of has electrical properties, but it also has chemical properties, and it's definitely biological. So Galvani showed, for example, that if you zap a freshly severed, freshly chopped off frog's leg with a little battery, which was super high tech back then to have invented a battery, it will twitch. And I'm going to show you that, I hope, on the next slide. So what we're talking about here, of course, is the fact that brains are communicators. Different cells in the brain, different regions of the brain have to communicate with each other. Brains are communicators. Okay, let's see if this took a while to load earlier today when I was testing it. There we go. And I hope it'll transmit for you. Should be hearing some music soon. Okay, I'm not going to watch the whole thing. Some people love that. Some people think it's totally gross. But you can see if you freshly severed that's most of the frog's body, which most of the frog didn't have a brain. The leg will move in response to an electric current, even without a brain. The leg doesn't know where the current came from. The leg follows the laws of chemistry and physics. And if you zap it with an electric current, much stronger, of course, than the one that the brain sends that way, the frog's leg is going to move. That was a huge discovery because it showed a sense in which the body and the nervous system follow the laws of chemistry and physics, not some magical spiritual rules that are impossible to put your finger on. So that was a big discovery. All right, let's keep moving. Let's try to. There we go. We really started to understand the brain really well, though, well after people like Galvani and Volta. It wasn't until the late 1880s, so about a decade after psychology had been invented, when this guy from Spain, Santiago Ramon y Cajal, hereafter I'm going to say Cajal because it's much easier to say, Santiago, who's a medical doctor and researcher, learned from other people. He did not invent this, but he learned from other people that you can stain brain cells. And if you do, you can get really good photographs of them. So by this point, photography was pretty highly developed. Microscopes were very highly developed. So he started developing systems to at least look at things through the microscope and then make very, very careful drawings of them. And his careful studies under the microscope helped us start to understand the physical structure at the microscopic level of the human and animal nervous system. So, for example, using these new staining techniques and experimenting with all kinds of them, Cajal very quickly discovered that all brain cells have a certain basic structure to them. They always have a cell body. They always have, it looks like just one axon, the sending part of the brain cell, and they have two, maybe a dozen, maybe 100, maybe 1,000 dendrites. These are the input signal. These are where the inputs are coming into the cell body. And so every neuron that he put under the microscope, and he looked at thousands of them, seemed to have that same basic structure. Now, they varied widely in different areas of the brain doing different jobs, but the basic structure was there. So just as cars and trucks and motorcycles vary widely, trucks are big, cars are small, some have two wheels, some have three. There are a few basic things that any motor vehicle is going to have, and Cajal discovered the same thing about nerve cells. There's a basic structure that shows up over and over and over. Okay. Notice what we're looking at here is the fact that nerve cells communicate with each other, so the axons and dendrites play that role. I hope you can also see here that Cajal started to discover that brain cells are really complicated, much more complicated even than he would have imagined. In fact, people debated for a long time exactly how complicated they were, and they kept drifting toward, wow, more complicated than we thought. Wow, even more complicated than we thought. Okay. It's also the case here that conservation is happening because if you move across different cells in a person's brain, or if you move from a human brain to a mouse brain, you see that bipolar neurons and Purkinje cells exist in both, and their structure is pretty similar in both. So Cajal notices hit 3 of these 6 cylinders. And of course, he recognized at some point that brains could be conscious, but back then computers didn't exist, so he didn't have a computer model to think about it. And of course, he also knew about compartmentalization, but he studied the brain at the microscopic level, not the more global level. All right, so that is Cajal. I've already said this a couple times and you read this, so I'm going to go quickly. Neurons are very conservative. They're remarkably similar in people and in worms or insects, for example. And in fact, even if you look at the electrochemical operation of a Venus flytrap, you may recall that it's one of about 500 plants that eat living things. So there are about 500 plants that are carnivorous. The Venus flytrap is just one of them. It's one that's native to North Carolina, at least some of them are. And so the Venus flytrap illustrates to us conservation. And before we talk about that more, let's see one at work. Okay, so we're going to talk about the conservative nature of the brain by looking at the Venus flytrap. Here we go. This is David Attenborough, I'm pretty sure.
The Venus flytrap.
They don't move that quickly, of course. This is rapid motion video.
It makes itself very attractive, oozing nectar across the brim of each leaf. visiting insect had better watch out for these 6 tiny hairs. This fly has to tread carefully. If it strikes one hair, it can carry on feeding, but a timer has been set. A second strike in less than 20 seconds, and the fly is doomed.
Now, wait a minute, I have to pause this because he just said something really cool. Some memory researchers would love me for saying this, and some would probably hate me, but from my perspective, he just told us that at least at a really, really primitive level, the Venus flytrap has something like a short-term memory. If a fly or other insect triggers a hair, no problem. But if the fly or insect triggers another hair, any other hair, I think there are 6 of them, in the next 20 seconds, it's going to close up. If it waits 30 seconds, maybe the fly's read up about Venus flytraps, it's going to be fine. So something at least vaguely analogous to a memory, not a long-term memory, only a simple short-term memory exists in the Venus flytrap, even though technically it doesn't have a nervous system. It certainly doesn't have a brain, but there's an analogy at least there. And of course, I'm going to tell you in just a minute that the Venus flytrap does all this stuff using a chemical that also is used by the human brain. It's called acetylcholine. But before we look at that, let's finish listening to, I'm pretty sure it's David Attenborough, the nature voiceover dude. All right, so let's watch this. I hate it when that happens.
An electrical impulse is triggered, and the leaf snaps shut in just a fraction of a second. The tips lock together like prison bars. If the fly is very big or very small, it may just manage to escape. But most are trapped and die.
All right. They make it dramatic, and it just gets sadder from here. You can go back and look it up and watch it yourself if you want. The link is right there, although you obviously can't click on it. But he just shows the demise of a few other flies and insects and makes the point that these Venus flytraps have evolved this amazing mechanism to make these very rapid movements. They don't have muscles. Their movement system is more hydraulic than muscular, but they operate based on this chemical called acetylcholine. And it's the same chemical that a mouse might use to move its whiskers, or I might use right now to pick up something that I'm going to use to teach this lecture. So acetylcholine, the neurotransmitter, plays much the same role in a carnivorous plant that it plays in the human brain, which I think is pretty cool. And of course, if I can get this to work, I have some difficulty. The conservation button should get active, and the communication button, of course, that's communication, chemical communication from where it's happening at that little trigger hair to the part of the plant that hydraulically closes up to capture the unwary insect. Okay, so conservative communicators. All right. We also know, I'm sure if you did the reading, you know this because it's a pretty big number and it kind of stands out. The human brain contains about 100 billion. Some people think it's only about 80 billion, and I'm sure it varies across people, but about 100 billion neurons. When I was in graduate school about 30 years ago, we thought it was only 10 billion. So we've been updating this number over the past few decades, but it seems to keep getting bigger and bigger. Though I should note that I think people now have their handle on a pretty good estimate. Obviously, you can't count every one of them, but I'd like to give you a sense, even beyond what I did in the reading, about just how big that number is. And right now, by the way, I'm only counting the basic neurons, not the helper cell. So just counting neurons, there are 100 billion of them in the human brain. How much is 100 billion? It's very hard for me to get my head around that, but I came up with a way to think about it just today that's a little bit different than what I put in the book. It's about one neuron, one brain cell for every dollar of Bill Gates's net worth. When I looked it up this morning, it was $109 billion. So, one neuron for about every dollar of Bill Gates's net worth. Jeff Bezos has a higher net worth. Okay, so Bill's not the richest man on the planet anymore. But to give you a better sense of how big that number is, if you wanted to convert $100 billion with a B to $100 bills, it would be 2,200,000 pounds of $100 bills. So it'd be many, many, many tons of $100 bills. That's a very, very, very big number. Okay, and I hope you will agree with me that that tells us that brains are very complicated. Something has that many little pieces, they fit together in a tricky, complicated way. That is a very complicated structure. Okay, let's keep moving. Now this overlaps with the book, so I'll go really quickly here, but if you also want to visualize or think about how many neurons there are in your brain by making them a bit bigger, you can ask yourself that question. And I hope you recall that if you had 100 billion ping pong balls, you would need 3 Empire State Buildings to hold them all. And that's taking out all the furniture. It's a completely empty building, and you got to fill it up. That's a lot. You also know that you could build a very, very, very big wall with them. You may recall that if you put them on a football field, you'd have to build a wall a mile high to hold them all. Okay, so it's really massive. It's a huge amount of space, and that just tells us that our brains are really, really complicated, really, really complex. Okay, to say a little bit more about the scale of the brain and the nature and scale of the neuron, the nerve cell, let's imagine you did blow up a motor neuron. A motor neuron is one of the larger neurons in the brain, I believe, and it certainly has very long axons. Some motor neurons have axons that are longer than a meter. They go from the base of your spine to your big toe, for example. That's one axon, one cell that takes a signal from your brain down through the spinal column into your toe so that you can wiggle your toe, for example. So if you took a typical motor neuron and you did blow it up to the size of a ping pong ball, the axon could be a quarter-mile long, okay, a quarter mile. All right, that's that. That's, of course, a crude drawing of an axon. And I hope you can answer this question for me. Why are the axons for a motor neuron so long? I sort of clued you into the answer when I talked about my big toe. The big toe is pretty far from the brain, 5 or 6 feet from the brain. And to get from the brain to the big toe, or even from the base of the spine to the big toe, you have to go a long distance. And it's possible that evolution could have invented a relay station with multiple neurons in the leg, but almost all your neurons are either in your brain or your eyes, by the way, a little bit of things like neurons in your heart, and of course, plenty in your spinal column, but there aren't any neurons. There aren't any cell bodies in your foot or your ankle or your calf. They're only in your nervous system that we think of as the brain and spinal column, spinal cord proper. All right, so that's that. And I hope you agree that that illustrates the complexity of the brain. Since I think I may have cut this lecture down a bit, let me just say briefly, as a quick reminder, what you see there in this little image here, those little yellow hot dog sausage shaped pieces, those are the myelination that helps a nerve signal, helps an axon transmit a nerve signal very, very quickly. Each individual one's called a Schwann cell, and each of those little golden hot dogs you see there, it's a fatty substance, that's a helper cell. Each of those little cells, they're called Schwann cells. It's a German word, Schwann. Each of those is a cell. It's even tinier, much tinier than the neuron, but it helps the neuron transmit a signal highly efficiently in a very rapid fashion. All right. So we see that neurons are conservative, they're complex. Of course, they're communicators. That's exactly what we're illustrating here. All three of those things are true. Okay. I got ahead of myself and mentioned that in addition to 100 billion neurons, there are about 100 billion helper cells, not just the Schwann cells, but several other kinds that keep things tidy, keep things moving, eat up dead cells. They do a variety of jobs. Some of them give nutritional support. So you all are probably young enough that you didn't watch Downton Abbey because you were only about 9 when it was a big show. But if you have watched that show, or at least heard about this show, maybe you saw the movie less than a year ago, I think it was. They do the helper cells to a wide variety of jobs, and without them, of course, the brain couldn't do its job. So another way which our brains are complicated is they don't just have neurons, they have an equal number of helper cells, and those helper cells come in a wide variety of structures and with a wide variety of functions. Okay, so again, things are complex. And if you want to look at this metaphor here in a little bit more detail, just as a British lord in 1910 couldn't do his job without a lot of support, your brain couldn't do its job without a lot of glial cells, a lot of support cells. And those are about equal in number. Okay, so brains are complex, right? We also know now that neurons do communicate with one another by means of a biochemical process. They're not levers. They don't operate on air or water or anything like that. They're not purely electrical. If they were purely electrical, nerve signals would travel at the speed of light, at the speed of electricity. That's 186,000 miles per second. They don't travel anywhere near that fast. They're fast, but not insanely fast. And we'll come back to that point in just a few minutes, I think. All right. I think I mentioned this before, but let me say it again. Every neuron in your brain, unless something really weird went wrong, has a cell body that's coded here in red. I circled it for you in red. An axon, just one of them, and you can see this is also an axon that has a myelin sheath. It's myelinated, so you have an axon. And notice in this case, as in many other cases, there are multiple dendrites. Dendrite, 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 dendrite. Lots. Oh, sorry, I skipped ahead too quickly. Another dendrite, and a couple of them, I guess, I didn't circle. Every nerve cell in your brain has a cell body, has just one axon, and it has anywhere between 1 and hundreds or thousands of dendrites. Okay, so that means that nerve cells are conserved. They all have the same basic structure. Nerve cells communicate. It's their job to do that. And if we wanted to, I forget if I did, we could certainly also circle complex. Let's see if I did about 3-4 months ago. No, I didn't. Okay, that's fine. Now, if neurons are going to communicate with one another, they have to solve a problem that Cajal noticed when he looked at them under the microscope, which is they don't touch each other. He looked and looked and looked and said, I've looked at this 1,000 times and I see under the microscope that one nerve cell never actually touches another. As I look at it, it's pretty clear that they're communicating, they're connecting to each other, but they don't actually touch. So what the heck is going on? The answer that he helped discover, that we now understand really well, is you have to have neural communication across a gap that's called the synapse. Okay, the synaptic gap, if you prefer. These are gaps that exist between the axon of one neuron, that's the sending part, and the dendrites of another that might be picking up on that signal, that are picking up on it, sending it to the cell body for processing, and then the cell decides whether to send an action potential of its own along its axon. So the gap is transmitted in this super tiny microscopic space called the synapse, and that means we need chemicals to help bridge that gap. It happens physically, not electrically, in that tiny synaptic gap. It only happens, of course, if there's an action potential. Okay, so if there's an action potential, an electrical signal, or at least quasi-electrical, goes across the axon, but at the end of the axon, where we have those things like synaptic boutons, you have to bridge that gap. It happens in an all or none fashion, and it happens through the release of neurotransmitters like acetylcholine, like dopamine, like oxytocin. We'll talk about a few of those today. And that neurotransmitter has to quickly, very quickly, bridge that super tiny microscopic gap, get picked up by the dendrites of the receiving neuron, and then register a signal, and then that neuron decides what it's going to do. Am I going to fire or not fire? And when am I going to do so? Okay, so that tells us that synaptic transmission is biochemical, not purely chemical, not purely electrical, and it's slow enough that we can measure it. And I think if you read ahead to the hands-on activity I probably put at the end, I think the end of module 5, you probably saw this, but I'm going to cover it for you anyway because it was in a part of the reading that might appear to be optional. Okay, so neurons are communicators. They're like computers because one neuron communicates with another and decisions get made to pick something up, to walk, to digest your food, that sort of thing. And I would like to demonstrate for you, if I can. Now, this is another one of those times that I'm sad to tell you we can't meet in person, because if we could meet in person, we could do a fun hands-on demonstration. But without meeting in person, we're going to sort of have to do this as a thought experiment instead of an actual demo. I would like to ask the question of whether it's possible without any high-tech equipment, okay, nothing fancy, but maybe a stopwatch, if we could prove that nerve transmission is not instant, it's not the speed of light, it takes time. It's somewhat slow, certainly relative to the speed of electricity. And so we're going to address the question of whether we could demonstrate that pretty easily without any incisions or microscopes or fancy equipment. And if I could, and of course I can't because it wouldn't be safe, I would take all 30 students in this course or another. I would take the 20 or 30 of you and line you up down a long hallway, as I've done many times. I would get you to stand up hand to hand or hand to shoulder, and we would measure the speed of neural transmission with a very simple stopwatch, no better than the one that you have on your smartphone. So here's what I would do. We're going to have to do it again as a thought experiment to illustrate that brains are communicators. It's going to look something like this, and I think this is the same image I put in your book. I may have tinkered with it a little bit, but notice here we have 8 people, and I just made them alternating red and blue so you can count them easily and see them. But notice in the top, that little cartoon or image there shows you if someone says go, the woman on, I'll point her out, the woman who's right here, okay, this woman, she squeezes the hand of her partner, and she squeezes the hand of her partner, and you work your way all along this very quickly. When the signal gets to the last person, I think that's Jeanette. Yeah, that's definitely Jeanette. Jeanette raises her hand very quickly, and I stop the stopwatch. So I would say ready, set, go, and I would start timing it with the stopwatch. The signal would move pretty quickly across a group of 8, 20, 30 people. It's a lot better if you have more people. And then I would measure it when Jeanette raises her hand. Now, it's the same group of people. So this is also Jeanette. That's Sarah, by the way. And so if we did it again, and we would practice this several times, so we get rid of practice effects and make sure everybody knows the instructions, everyone's paying attention. If we did it again, the way I'm showing it to you right here, I'd like you to stop and think with me a bit. Would it be any different? You still had to transmit the signal from one person to the next, to the next, to the next, to the next, and so forth, down to Jeanette. But would it take, now again, we're accounting for practice. So of course, as you do this more, you get faster. We give people plenty of practice. We take care of that. We mix up the order in which they do the activities. But so ignoring any effects of practice, holding that constant, would you expect people to be any quicker, any slower, or exactly the same speed to transmit a message from one person to the other person? I'll give you a few seconds to think about that. if you didn't read ahead and read that. If you read, you already know. All right, I wish I could call on somebody here, but I hope you drew the conclusion or remembered that in the second case down here, in the bottom activity, remember, same group of people, you don't need random assignment, by the way, if you use the same people as their control. So if this is Jeanette, this is still Jeanette. I think I said that was Sandra. If that's Sandra, it's still Sandra. Okay, that's still Lakeisha, that's Lakeisha. The 8 people didn't change. They didn't get older, they didn't get smarter, they didn't get sillier. It's exactly the same 8 people. So this is a within subjects experiment, of course. I've done it now dozens of times at lots of colleges and universities, and I always find that it's faster in the lower case, the second case down here that I'm pointing out for you, because notice, if I track, I hope you can see this in Screen-O-Matic, if you track how far the signal has to go, it's got to go from her brain down this arm, up that arm, into her brain. Think about it, do it quickly, across that arm, across the rest of the arm, and it keeps going. But notice that we cut an arm out of the picture right here. So it had to travel, let's say about, I don't know, 2 feet, 2.5 feet less in this example. So we can cut out 2.5 feet, 2.5 feet, 2.5 feet. Oh wait, this would be 3 feet because it's a very tall person, only about 2 feet because it's a very short person. We would cut out about 30 inches or so as we worked our way into the second situation here. So when I've done this, I've found very consistently that it's quicker, measurably quicker in the second case than it is in the first case, which is a pretty fun way to see. Yeah, nerve transmission takes time. In fact, I've even compared my calculations a couple of times to what the research literature says, and we usually come pretty close to what we think is the speed of that kind of nerve transmission, because nerves travel at different rates in different parts of your body and different parts of your brain. Okay, that's a student text me, but I'll have to get back to them later. All right, so that's that. I wish we could do it in person, maybe in a future world, but that's that. And that, of course, also illustrates that brains and nervous systems, of course, are communicators. It's their main job to transmit information. Okay, I forgot that our break came so quickly, but it's here. That takes us about 37 minutes. So I'm going to take a break here and shore up this lecture. And as soon as I can get this one posted, or starting to get posted, I'll go to work on the next lecture. All right, take care.